How you doing, guys? Welcome to another video. This is option B, high level biochemistry, nucleic acids. Let's go. Okay, so volume 8, nucleic acids. We talk about nucleotides, we want to know what they are. We talk about base pairing, and then DNA synthesis. The IB understandings and applications and skills are quite long for this section, so make sure you check out B8 in the guide. But mainly in this video, we're going to focus on the structure and on the chemistry and the bonds that link them together. I doubt they'll ask you to draw a section. So DNA, that is a condensation polymer constructed from 5 monomers known as nucleotides. Each nucleotide consists of a phosphate group, a deoxyribose sugar, and one of the 4 bases, either A, G, C, or T. Sometimes we use U if we're looking at uracil, and we'll talk about that later. Each of those 5 bases can be found in the data book, so you don't need to remember the structure. So if they're talking about DNA, refer to the data book. Adenine, guanine, and cytosine can be found in both DNA and RNA. Whereas thiamine and uracil, well, they can only be found in DNA for thiamine and RNA for uracil. They swap each other out. Now with the data book, you need to be able to identify the link where the base joins to the sugar group. So I've gone through and I've highlighted the hydrogen that's responsible for creating the covalent bond between the sugar molecule and the base. Sometimes we'll need to reorientate it or twist it around to get that bond to look correct, but if you can identify which hydrogen makes that connection with the base, you'll be in a good situation if asked to draw a section of a nucleotide or a section of the DNA chain. So that's how they're presented in the data book, so make sure you can identify the right location. The easiest way to remember about a nucleotide in DNA is phosphate, sugar, base. Phosphate, sugar, base. Phosphate, sugar, base. And each of these connections is a covalent bond where we have eliminated a water molecule. That's why it's referred to as condensation polymerization. This box that I've circled on our phosphate sugar base, that is known as a nucleotide. So if they ask you to draw the nucleotide must have a phosphate, a sugar, and a base. A nucleoside is just a sugar and a base. So make sure you know the difference between those two terms. A nucleotide is made up of 3 components: our deoxyribose sugar, our phosphate group, and one of the nitrogenous bases. Those 3 components are linked via covalent bonds formed from condensation reactions. The base forms a covalent bond to the carbon of the sugar labelled C1. So C1 is the carbon on the right hand side of the deoxyribose sugar, and that is the location of the base. The phosphate group binds to the atom labelled C5, and that is if we were looking at one above. So there's a phosphate group attached above. The other phosphate group would be bonded at the 3 location to keep the chain going. So here's a little bit of a representation of how we would come up with the nucleotide. We have our phosphate group, which is attached to our number 5 carbon, and then we have our base, which we would need to orientate so that that hydrogen that makes the connection is attracted to the hydroxide functional group. You can see there that we will eliminate water, and that's how our link is formed. The link between the sugar and the phosphate group is sometimes referred to as a phosphodiester link because it contains the ester functional group, but instead of having a carbon, it's attached to a phosphorus. The link that joins the sugar to the base is simply a covalent bond between the carbon and the nitrogen. It's not given a specific name, but it's just a strong covalent bond that is formed from a condensation reaction. All of the other bases would do the same type of reaction. We just have to make sure we get the orientation correct. So DNA is a polymer made up of the nucleotides. We join the nucleotides together to make up DNA. DNA will always consist of our pentose sugar, our nitrogenous base, and our phosphate group. They're joined together, the sugar and the phosphate group, by an ester link formed from a condensation reaction, and then the different nucleosides are due to the different bases. So here we have an adenine nucleotide where we can see all of the things joined together. So we've got our phosphate group, which is connected to our sugar, and here I've identified the ester link or the phosphodiester link. Here I've identified the covalent bond between the sugar and the adenine base. So make sure you can identify those parts and name them as well. So once we've got our nucleotides, we can link those together via condensation reactions between a sugar and another phosphate group, building up the DNA chain. So we keep on removing water, and the sugar of one of the nucleotides connects to the phosphate group of the other nucleotide, and then in those phosphodiester links, we're getting our backbone of DNA to be established. So here I have my first nucleotide that contains an adenine, our second nucleotide, a thymine, and our third nucleotide, a guanine. I'm just labeling the carbons so you can see at which locations the number 1 carbon, that's got the base, the number 5 carbon, that's got the phosphate group, and our number 3 carbon is what will connect to the phosphate group below. Each of those reactions will produce water, so they're all condensation reactions. What we'll do is we'll form our backbone of DNA, which contains our phosphodiester links, and now all of the nucleotides are connected together. So we're starting to build up the DNA chain. Remember, if they ask you to highlight or circle or name those bonds, the bond between the phosphate group and the sugar is an ester or a phosphodiester, and the bond between the sugar and the base is a covalent bond. So RNA and DNA are both condensation polymers formed from nucleotides. DNA is a phosphate deoxyribose sugar and a base. Now the deoxyribose does not contain an oxygen atom on the second carbon. An RNA nucleotide is a phosphate group, a ribose sugar and a base. And a ribose sugar has a hydroxide functional group on C2, and that's the only way they differ. In terms of their structure, the RNA will swap out thiamine for uracil. So there's no thiamine in an RNA molecule, only uracil. So the uracil will take the place of thiamine. How do these things function? Well, DNA is known as a double helix, so it's double-stranded. Both of those strands are running in what we call anti-parallel to each other. RNA is a single-stranded molecule. Now, what is the primary and secondary structure of DNA? So the DNA molecule is double-stranded and it runs in a direction called anti-parallel. That means one's going up like that and one's coming down like this. And in that anti-parallel arrangement, we form primary and secondary structures. Now the primary structure is easy. It's essentially what bases are attached to the phosphate and the sugar. The secondary structure has to do with the bases, and it's the hydrogen bonds between base pairs. So A and T are a base pair, and they contain 2 hydrogen bonds between both of those molecules. G and C are another base pair, and they contain 3 hydrogen bonds between those molecules. That hydrogen bonding creates the secondary structure of DNA, and it also has impacts on how the DNA functions. So again, that anti-parallel, if we have the 3 sugar at the top of one, then coming the other way, we'll have the number 5 sugar. So anti-parallel means one running that way, running that way, and then we have our hydrogen bonding in between our bases, which keeps the molecule together. Taking a closer look at the base pairing between A and T and G and C, we may need to describe the location of the hydrogen bonds between the base pairs. So here I have the base pairs of A and T and G and C, and we're looking for the location of O bond H or N bond H. There's no fluorine in any of those bases, so we don't have to consider that. So here we have our adenine and our thymine, and we know that that will have 2 hydrogen bonds. So the N bond H is attracted to the lone pairs on the neighbouring oxygen, and vice versa for the second location. G and C, well, it has 3 locations for hydrogen bonding. Will they ask you to draw those 2 things and label them? That would be an incredibly hard question. I think they're more likely to ask you to indicate the location. If we have a strand of DNA that contains more of G and C, then that's going to be harder to separate due to the larger degree of hydrogen bonding. The tertiary structure is the interaction of DNA with these molecules called histones. Histones are proteins, and what happens is the DNA coils itself or supercoils itself around the histones, which essentially create our chromosomes. DNA is negatively charged, and the positive charge of the histones allows the interaction. Now, DNA replication. When we have replication of DNA, the 2 strands unwind, or they're unzipped. They are unzipped by an enzyme, which allows us to make an exact copy of the strands. So what happens is the polymerase partially unzips part of the chain, and then we form a template. So if we have an adenine base, then what will happen is the enzyme will go and find a nucleotide containing the thymine, and then it will come and arrange itself next to it. So in this process, we get 2 exact copies of the original DNA by the free nucleotides joining into the chain. So DNA is the genetic material that an individual inherits from its parents. RNA is directed by DNA, but RNA is responsible for protein synthesis. And protein synthesis occurs via a triplet code. The way this process occurs is DNA is in the nucleus, and RNA needs to be transcribed. So there's a transcription process that occurs in the nucleus that releases RNA so that it can leave the nucleus. Once it leaves the nucleus, we have a template for which we can start to build our proteins. So once the RNA is out of the nucleus, we have this compound called transfer RNA, which starts to collect the amino acids that correspond to the base pairs. And the base pairs come in a triplet. So for instance, GGA, that triplet refers to an arginine amino acid. So it will go and collect that amino acid and place it in the peptide chain. And then ACU, well, that corresponds to a different amino acid. So the transfer RNA goes and collects that amino acid and places it next to the arginine. You can see that the peptide chain can be built up by following that triplet code. And the longer the protein gets, the more amino acids have been collected. The final thing we need to talk about is GMO, or genetically modified organisms. These are where we have taken the DNA of one species and spliced it or interchanged it with another. So for instance, corn, that is a GMO product, and some rices are GMO products as well. And we need to be able to identify some of the benefits and concerns. So some of the benefits with GMO food might be a longer shelf life. Some of them, where they've created a natural pesticide to go on the outside of the fruit or the food, it means it's got an increased resistance to disease. And another good thing about GMO foods is maybe we can increase the yield. So farmers will be able to produce more using the same amount of land. Some of the concerns people have with GMO food is that there are no long-term studies about the effects. So we don't know the effects over a long period of time. Some of the other ones is that GMOs have been linked to allergies and young people forming more allergies to certain foods. For example, if you're allergic to fish and there's fish DNA in a certain food, then there's a chance you'll have a reaction. And finally, the last concern is that we've changed the natural food composition and we've altered that process, and how that impacts nutrition and all those other things are important to understand. So Volume 8, some top tips. Remember the sugar phosphate base. Sugar phosphate base. That will get you through so many questions. Remember the number of hydrogen bonds between A and T and G and C. And remember you've got U instead of T in RNA. Thanks for watching, guys. Don't forget, drop a like on the video, subscribe if you're new, and I'll see you next time.